Hey guys, welcome to this revision video on brain scans and the brain scanning research that you need to know for the GCSE. So what we're going to run through in this video are the 3 brain scans, the CT scans, the PET scans and the fMRI scans. A quick bit of evaluation for each of them, maybe how to answer some questions along the way, and we'll have a little look at the key study, Tulving's Gold Memory Study. So this entire part of the GCSE is learning about what functions, what areas of the brain have what functions, lining up brain functions to specific parts of the brain. And as we mentioned in the last video, this is called cognitive neuroscience. Now, what we can do in modern society with the technological advancements is we can essentially investigate a person's brain while they complete different cognitive activities by putting them under a brain scanner. And what we can look for is we can look for brain areas that are active, brain areas that aren't lighting up, because that tells us that that function that they are doing is linked to that part of the brain. So the first scanning technique that you need to know about is called the CT scan. And the CT scan, it basically works by taking X-ray photograph slices of the brain, and what we will do is we will put these slices together to create quite a detailed picture of the brain. Now, whilst these are photographs and they're not live images, so what we can't do is directly see what part of the brain is active at that precise moment in time, what we typically use them for with psychology is we will use them to identify a function that a person cannot complete, let's say speech production, and we will look for brain damage that clearly lights up or is clearly displayed on the CT scanner. And we can make that little link there, that that brain area that is damaged must be responsible for the function that that person can't do. So in terms of how you might describe that for an exam, if you had a question like this, describe how a CT scan is used to look at brain functioning. Basically, you could say something like, CT scans take X-ray images of slices of the brain, which are put together to make an overview of a person's brain. If a person cannot complete a cognitive task, we can look for a damaged brain area in the CT scan and confirm that this brain area is responsible for that function. You need to be able to evaluate the CT scan, so what we could say on the positives is that CT scans are very clear. It's very obvious where the damage is. So at a higher level, we would say that this means it's got very high spatial resolution. On the downside, lots of X-rays does mean radiation, so too many of these could become a harm to the patient, which is a problem. If you're just going to the hospital for a standard CT scan, though, you've got nothing to worry about. The other negative is that CT scans are not live, so we cannot use this technique to scan the brain in action live. So at a higher level, we would say this has low temporal resolution. We can't see it live. Again, if you're going to get a question for evaluating it, you've got to remember the 2-step process of each evaluation. You've got to identify an evaluation and then explain it. So you might say something like, CT scans create very clear images. This is good because it means that we can see exactly where brain damage is and link it back to brain functions. Okay, the second brain scanning technique that you need to know is the PET scan. PET scan is a lot more modern than a CT scan, and it works slightly differently. Before a PET scan, you have to inject the patient with a small radioactive tracer that goes into the bloodstream, and the PET scan will essentially then measure the activity of the brain. And what it does is it measures the concentration of the radiation in the bloodstream. So because the most active brain areas absorb the most blood, the PET scanner will identify and monitor where the most radiation is at that moment in time. And it basically will send that to a computer screen showing it in like blue, yellow or red colours for the most active areas. The more active an area is, the more blood and radiation will be absorbed. So what we can do is we can put someone under a brain scanner, PET scanner in this case, ask them to do a task and we can observe where the blood and radiation is being absorbed. Similar sort of question, if you had how to describe a PET scan, you would say something like, PET scans measure activity in the brain. A patient is injected with a radioactive tracer, and the PET scan measures which brain areas are using the most blood. The areas that use the most blood will show up red on the screen, and we can ask a patient to complete a cognitive task and see what brain area lights up. Evaluation-wise, this time we can use PET scans to test the brain live. So we could say that we can see the brain in action, it's lighting up very quickly. This means it's got high temporal resolution. It's also, again, a very clear brain scanning technique. We can identify very clearly where the brain function is, where the blood and radiation is being absorbed. So we could say high spatial resolution again. It does involve radiation injections though, so again, there is a risk of long-term harm, and they are a very expensive process. So you can't really afford to do lots of them for research, which often means you end up with quite a small sample. Again, evaluating, remember the two-step evaluation process. Something like PET scans create live images of the brain. Excellent. Now tell me why this is an evaluation. This is good because it means that we can see exactly what brain areas light up when doing a cognitive task, so we can confirm brain functioning. The final brain scan technique is the fMRI scans. These are different again. These are machines that use giant magnets to take constant photos and images of the brain to see the levels of blood and oxygen in the brain. It uses giant magnets because it is able to track the level of blood, and what we are looking for is we're looking to track the amount of oxygenated blood. So the more oxygen that is required for a brain function, the brighter that area in the brain will light up. So the more active and more red it will be. So for example, if we were using the visual cortex, we would see a lot of oxygenated blood go to the back of the brain. Similar to the PET scan, we can ask the patient to complete a task under a brain scanner here, and we can observe which brain areas are lighting up. But as you can tell, this is a very different brain scanner because it uses giant magnets and no radiation. So if you were going to explain how it works, you could say something like, fMRI scans measure activity in the brain. They use giant magnets to track the location of blood in the brain. When a brain is active, it will absorb more blood and show up red on the screen. We can ask a patient to complete a cognitive task and see what brain area lights up with the most blood. Evaluation-wise, once again, we can say that it's a very clear process. We can see exactly what part of the brain has lit up. So that's very obvious for the researcher to know where the brain function is. So high spatial resolution. We can say that they're using magnets only, so there's no radiation and no injection. So it's the safest brain scan. So it's non-invasive. We can say it's very expensive, which again means you're not going to be able to run lots of experiments. So you're going to get a lower sample. And if you've ever had an MRI scan, they're very loud, they're very claustrophobic, and they require you to stay very still. So that makes it quite difficult for people with extra needs or children to complete this brain scan. So it makes it difficult for certain people. Again, remember the two-step evaluation process. So something like fMRI scans create live images of the brain. This is good because it means that we can see exactly what brain areas light up when doing a cognitive task, so we can confirm brain functioning. Now, the last thing you need to know is the key study to this little section, and it's Tulving's gold memory study. And what Tulving wanted to do was to see what brain areas were responsible for memory. So what Tulving did is he got 6 volunteers and he injected them with a radioactive gold tracer. That's where the study gets its gold name from. He then PET scanned them and asked them to think about episodic memories. He then repeated the experiment with the injection, put them under the PET scanner again, and asked them to think about semantic memories. The PET scanner basically was just monitoring the blood in the brain to see what parts of the brain were more active. The first interesting point for evaluation is that 3 participants were dropped due to inconsistent results, leaving us with just 3 participants left. But in these 3 participants, when they were thinking about episodic memories, there was greater blood flow in the frontal lobe. And when participants were thinking about semantic memories, there was greater blood flow in the parietal lobe, showing us that those 2 types of memories are in different brain areas. So that leads to the conclusion that episodic and semantic memories are clearly located in different parts of the brain. You need to know this study in case you get asked to describe it in the exam. Now, you could get asked to evaluate it. So the evaluation's nice and straightforward. First one is clearly a problem. We finished with only 3 participants. They were volunteers. One of them was himself and another one was his wife. So not only is this a small sample, there's quite a risk of bias. As it was a controlled lab brain scan process, we can say that we should be having quite good control of extraneous variables. We should be able to replicate this study in the future. So that's a good thing. However, as always, a controlled lab setting comes with the risk of unnatural behaviour. And finally here, in a brain scanner, asking somebody to think about specific things, it's actually impossible to know that they were thinking of the exact thing you asked them to when you asked them to think about it. So there's always that risk that they were thinking about the wrong thing. So the brain scanner was picking up the wrong information, so that they may have inaccurate conclusions and they may be making the wrong conclusions about what part of the brain is responsible for episodic and semantic memories. So there's always that risk, which is a bit of a bad thing. So guys, this concludes the video here on the 3 main brain scans that you need to know for the GCSE, as well as Tulving's Gold Memory Study.